Hi everyone. In this video, we will dive into the smallest unit of life, the cells, and understand the organelles within them. So primarily, there are uh, two ways to classify cells. The first one is dependent on cell morphology, which is basically any shape and features that go along with these cells. And the first type of cell based on morphology is archaea, or not archaea, prokaryotes. These basically do not have a membrane-bound nucleus. And then we have eukaryotes, which have a membrane-bound nucleus. So the main difference is the presence of a nucleus or not. Now, according to phylogeny, there are three groups of living organisms. The first one is bacteria, where they are unicellular prokaryote cells. Then we have archaea, which are also unicellular prokaryotic cells. And then we have eukaryotes, which can be unicellular or multicellular. And if you see these, you see different types of life aspects or species, that kind of thing. So in a prokaryotic cell, what we see is they have no membrane-bound nucleus. They vary in shape and size, but contain several structural similarities where they have a plasma membrane, a single circular chromosome in the nucleoid region right here, where it also contains maybe small plasmids, and then which are like small uh, particles of DNA or nucleic acids. Then there are ribosomes, which ends up synthesizing the proteins. And then there's a thick, stiff cell wall that keeps everything uh, contained. So prokaryotic cells also have some internal structures. In addition to the nucleoid, the chromosomes and the plasmids, the again there are ribosomes but no endoplasmic reticulum. And then there's many prokaryotes which have internal photosynthetic membranes because they need to create energy, which we'll talk about later on. And then some prokaryotes also have a membrane that's protein enclosed organelles or microcompartments. So like tiny compartments where other aspects of the cell is enclosed. And then many of these cells also are supported by a cytoskeleton of long, thin protein filaments. So with prokaryotes, there are also some external structures. Some of them have a tail-like flagella. So that's like this structure here, where it allows it to spin around, move the cell by spinning around. And then with most prokaryotes, they have a cell wall that surrounds the plasma membrane. And then there's another layer of outside the cell wall that is just basically with glycolipids. Now we branch into the realm of eukaryotes. Um, these can range from like algae to like 100-meter tall redwood tree, like this tree here. Many of them are multicellular, but they're like plants and animals, but there are some that are unicellular, like protists and some algae. So eukaryotes have a very large surface to volume ratio compared to um, prokaryotes. This we've seen in like previous pictures where we have uh, the scale bars being almost 4 times the size in terms of like the cell itself. And with eukaryotes, the overall rate of diffusion is very slow for the speed of life. So cells end up breaking up the large cell volume into several smaller components called organelles. And this compartmentalization allows these cells to have two primary advantages. It allows the separation of certain chemical reactions that are incompatible with each other, and it increases the efficiency of certain um, chemical reactions, for example, chloroplasts, which are involved in photosynthesis, and we'll talk about those more later on. So this is an example of a eukaryotic cell where it's a plant cell. Plant cells also have a cell wall compared to other ones and chloroplasts and all that. Make sure you know what each components are. Like if one is labeled like, what is this structure here? You should be able to recognize as the Golgi apparatus. Or if it's like this, it should be like the chloroplast, depending on the question itself. These two are specific to plant cells, not animal cells. So the cell wall and the chloroplast. And then with eukaryotic cells that are animal cells, uh, most of them are seen within plant cells except for centrioles. Now, between the two different cells, when we look at them, they both have microtubule organizing centers. But animal cells also have centrioles that are associated with this center uh, in this complex that is called a centrosome, and this is absent in plants. And within plant cells, they have a cell wall, chloroplasts, and other specialized plastids, and a large central vacuole. Um, in most cases, scientists end up classifying the plant cells as a vacuole, plant vacuole as a type of lysosome. So we have these different structures. Make sure you guys know the differences between plant cells and animal cells. And now it brings us to the first organelle, which differentiates between prokaryotes and eukaryotes, and that is the nucleus. So the nucleus is a highly organized large organelle, which is basically has the structure of a double membrane nuclear envelope with nuclear pores that end up regulating traffic in between, like these little pores here. And what happens is that the ribosome production occurs within this nucleolus area itself. and it functions as information storage and processing for chromosomes and ribosomal RNA synthesis and the production of ribosomes within that nucleolus area. So there are chromosomes around it where and in there we have the nucleolus where the ribosomes are produced. Now, I keep saying the organelle ribosomes. What are they? What do they do? So ribosomes are organelles that are primarily functioning in protein synthesis. They can be either free in the cytosol, or they can be in the rough endoplasmic reticulum. And these are basically little non-membranous little structures that contain RNA molecules and proteins. They are technically not considered organelles, but it's within the cell to assist with protein synthesis. So now we have the rough endoplasmic reticulum, or the RER. This is a network of membrane-bound tubes and sacs with ribosomes attached. And it also has an interior that's like a lumen, and the rough ER is continuous with the nuclear envelope itself. And what this component of the cell does is that it allows protein synthesis to occur at the ribosomes associated with the rough ER. And then these new proteins are folded and processed in the lumen of this rough ER. And what happens is that protein containing vesicles end up coming off the rough ER and travels into the smooth ER or the Golgi for like transport to other places in the cell. Now we have the smooth ER. The smooth ER is similar to the rough ER, where they are the endoplasmic reticulum without ribosomes. And what these smooth ERs do is that the enzymes within this smooth ER's lumen ends up synthesizing different fatty acids and phospholipids, and or break down any poisonous lipids that entered the cell itself. And then it also acts as a reservoir for calcium ions within certain cell types. Now we have the Golgi apparatus. This is basically formed by a series of stacked flat membranous sacs called cisternae. So like these here. And these also are like vesicles that carry new proteins and butt off from each other like this, as like little secretory vesicles coming off like these transport vesicles, and travel from the nucleus towards the cell membrane, that kind of direction. And what happens is, what the Golgi apparatus does is that it processes, sorts, and ships proteins synthesized within the rough ER and membranous vesicles from the Golgi apparatus ends up carrying the sorted material from the Golgi to wherever they are supposed to be. So now protein transport itself, it has proteins themselves have a specific destination. When it when it's being secreted. So when there is like a signal sequence with the amino acids, that's the primary structure of this protein. What happens is that it allows them to be sorted out and packaged into transport vesicles and gets directed by different microtubule tracts to get them to where they belong through like motor proteins and other aspects to kind of guide them along the way. Then the endomembrane system itself is a continuous integrated system uh, that is composed of the nuclear membrane, the rough ER, the smooth ER, the Golgi vesicles, and the lysosomes. And these proteins and lipids and membranes, they are all moved through the system and transported through the destination. So they all move around to get everything where they are actually supposed to be. So now we have a structure called peroxisomes. These are globular organelles that have a crystalline core with a single membrane, and the crystalline core itself contains many types of enzymes that are involved in the breakdown through enzyme catalyzed oxidation reactions and reactions that need to be compartmentalized to avoid cellular damage. Now, lysosomes, on the other hand, they are also single membrane bound structures that contain approximately 40 different digestive enzymes, and they are basically used in digestion and waste processing of the recycling of cell components. So basically, it's like the area where it kind of sorts out all the toxins and all the different waste particles, and it sorts it all out and then recycles some aspects for like cellular components. Now there is part where it's called endocytosis. This is basically a way to bring in materials into the cell. The materials are delivered by lysosomes for degradation and recycling of these processes. And some of these forms of endocytosis are called phagocytosis, which means like cell eating. Penocytosis is like cell drinking. Autophagy is like self-eating, or just like a little self-destruct button. And then receptor mediated endocytosis, which basically selectively takes up particles that are around the cell itself. Now, there is also a process called exocytosis, where proteins and other substances are destined to be secreted from the cell and transported in vesicles like this out and out, and it goes and fuses with the membrane, and it goes exiting out of the cell itself like this. That's called exocytosis. Now there are structures called vacuoles, which are basically large membrane bound structures that contain some digestive enzymes. These are particularly specialized for digestion and nutrient storage. Most vacuoles are used in storage of water or ions to keep the cell to maintain its normal volume and like the size itself. And if there's like not enough water inside the cell, it goes hypertonic. If it's has too much water inside, what happens is it goes hypotonic, because the water is coming in so much, and in this case the water is leaving the cell so much that they're in a situation where they're in like salt areas outside. We'll talk about more of these in the next video. And this is just equal amounts, water inside and outside. So now we have the mitochondria and the chloroplasts. Both of them do aspects of cellular respiration. So for animal cells, this is more for cellular respiration, whereas chloroplasts are particularly for photosynthesis. It also, with mitochondria, they also produce ATP for like energy. And there is a theory called the endosymbiont theory, where mitochondria and chloroplasts are former prokaryotic cells that were incorporated into the eukaryotic cells. And if you want to know more about that, I would check out this video as well. So now there's the cell wall, where it's basically an outer rigid shell wall, where in plants and algae, its primary component is cellulose, and then the primary component when it's fungi, it's chitin, and it basically is there just to protect the cell, help it keep its shape, and then regulate what goes in and out of that cell wall itself. Next, there's also the cytoskeleton. It's a highly dynamic structure in that it also changes to alter the cell's shape to provide cells with necessary components to transport different materials or to allow mobility of the cell itself. The cytoskeleton itself is composed of different protein fibers. These include the microtubules, actin filaments or microfilaments, and intermediate filaments. So these are particularly, the microtubules are particularly focused on movement of the cell, using like motor proteins and ATP. The actin filaments, it gives the cell shape and allows the entire cell to move. So microtubules are movement within the cell, like components within the cell. And then actin is the whole cell moving places. And then with intermediate filaments, what we see is that there is cell structure and support. Those are the primary functions where it keeps that shape itself. Now with microtubules, what we see is that there are large hollow tubes made of tubulin dimers like this, where tubules can grow and shrink by adding and removing certain tubulin units, and originate from the microtubule organizing center or centrosome in animal cells, and acts as a railroad to move the transport vesicles or the chromosomes within like a cell. So it allows it to, it's like taking a step throughout. It just walks on it kind of thing. And essentially the motor proteins here end up using ATP to transport any vesicles along these microtubules. So they're just walking away. Now the actin filaments, these are the smallest of the cytoskeletal elements. These are formed by polymerized actin molecules. It helps define the shape itself. It involves the movement of the entire cell or the cytoplasm with interaction with the motor protein myosin. So actin and myosin, they're like best friends. They want to go together. And actin myosin interactions, what leads to them is that they end up causing cell movements such as cell crawling, cytokinesis, and cytoplasmic streaming. So that's what this is. So these are some examples of how they interact, and what actions they play. And then intermediate filaments, those are defined by size rather than composition. There are many types, each consisting of different protein. And what these intermediate filaments do is provide structural support. They are not involved in movement like the other two sets of cytoskeletal elements. These intermediate filaments end up forming a flexible skeleton that allows the cell to form that shape and hold that nucleus in place where it's supposed to be. Now we have cilia and flagella. It allows the entire cell to move around, and these are so flagella itself are long hair-like projections from the cell surface that move cells. The bacterial ones are made of flagellin and rotate like a propeller, whereas eukaryotic ones are made by microtubules, which goes back and forth rather than rotating. And then cilia, they are short filament-like structures like these, which basically move like oars to a rowboat and generate fluid current to move the cell itself. And cells may have either one or two flagella, but have many different. So it could be like one or two of these, and then a million of these, if it's necessary, depending on the creature themselves. And with that, we will continue in the next video in regards to the plasma membrane, how that structure works, and have some fun with ideas related to the plasma membrane.